Hey there everyone, we are from Group 5 and we will be discussing on the overview of robust methodology of ADC production for anti-cancer treatments. As we all know, cancer is a leading cause of death worldwide, accounting to almost 10 million deaths in the year of 2020 itself. Current available treatments such as chemotherapy and radiotherapy are used in tackling cancer, and those treatments involve the use of strong chemicals that destroy fast-growing cells. In these treatments, patients experience fatigue, depressive episodes, pains, nerve damage, cardiovascular issues, and changes in cognitive functions. Such a significant issue resulted in the need for a diversified cancer treatment option that does not cause as many drawbacks towards the patient's health. Antibody-drug conjugate or ADC is an alternative method for cancer treatment. It is made of a monoclonal antibody, a linker, and a cytotoxic payload. It works by taking advantage of the different features of the cells in the tumor. ADCs work by sending covalently bonded cytosis molecules to the tumor cells that duplicates the targeted antigen to new normal cells. Optimized ADC production uses Chinese hamster ovary cells or CHO cells, as these cells have successfully shown that they could bind to breast cancer cells. Several other cells can be used for the production of ADCs, but only some of them are approved for the production of ADCs, and only some of them can be used to produce ADC at a large scale. CHO cells are used because they are known for their stability that is considered advantageous for the safety in human application. This is a stirred tank bioreactor that is set up in a fed batch mode. Large-scale ADC production using CHO is produced here. Fed batch mode is where one or more nutrients are fed to the bioreactor during cultivation and in which the product remains in the bioreactor until the end of the run. It is used because its productivity is 6 times more efficient than simple batch operation. There are several parameters to this fermentation process, for example, having a 7.3 to 7.5 pH to cultivate the CHO cells. Next, a temperature of 37 degrees Celsius, which is the optimal temperature for cell growth, since its enzymatic activity and metabolic activity are dependent on the optimal temperature. Next, a dissolved oxygen or DO of 70% and a rate of agitation of 70 rpm. The product yield of CHO cell cultivation is about 0.135 grams per day in a stirred tank bioreactor. On the other hand, the product yield of MAB is about 1.9 gram in a stirred tank bioreactor. All of these data was taken in a course of 49 days. The previously mentioned process is called the upstream production, which ends when the MAB cell lines have been fermented in the bioreactors. Next, the downstream process of the MAB is divided into 3 parts, which are harvesting, purification, and polishing. The first step is harvesting. The main goal of harvesting is to obtain HCCF or harvested cell culture fluid, which is when the yielded cells or cell debris is suitable enough for chromatography. Harvesting first starts with tangential flow filtration or TFF, which works when the HCCF flows in the tangential direction to the microporous membrane and the insoluble cells are separated by a pressure that is driven by the filtrate flow. Next is the centrifugation process, which is done simultaneously with depth filtration for a very long time. Lastly, the subject clearance process, which is the step that guarantees the product safety and high efficacy rate. Next is purification. Purification mainly consists of chromatography and flocculation. We are going to first talk about chromatography. A popular purification technique is chromatography because of its high resolution technique. A type of chromatography that is known for its high level of purity is protein A-based chromatography, which is also mostly used for the production of MAB. Next is flocculation. The flocculant in this process is chitosan, which is found through fine purification efficiency due to its ability to flocculate submicron particles and cell debris, which usually escape the sedimentation in the centrifugation process. Last is polishing, which is basically just an addition of chromatography steps, either ion exchange chromatography, hydrophobic interaction chromatography, or hydroxyapatite chromatography, or mixed mode chromatography. In every production process, challenges will always exist. In this case, one of the challenges is when cancer cells undergo therapeutic stresses, which causes them to develop resistance to survive the cancer treatment.
Created using Powtoon.